Today we're interviewing the authors of High-throughput protein crystallography to empower natural product-based drug discovery. It's a paper that was recently published in our journal, and the authors are all from Brazil, starting with the lead author, Raphael.
Hello everyone, I'm Raphael. I'm a specialist in technological development here at the Brazilian Center for Research in Energy Materials, CNPEM. I work at the Division of Drug Discovery led by Daniela as a crystallographer, and my role in this work was to do the whole crystallography experiment from crystallization, data collection, and data analysis using our high-throughput crystallography pipeline.
Thank you very much. Next, we've got Andrey.
Hello, everyone. I am Andrey Nascimento from Brazilian Synchrotron Light Laboratory. I am responsible for the MANACA beamline, the beamline for macromolecular crystallography at Sirius. I have been responsible mainly for the data processing development for this paper.
Brilliant. Thank you very much. And finally, we've got Daniela.
Hello, everyone. I'm Daniela Trivella. I am the head of drug discovery here at CNPEM, at LNBio, the Brazilian Bioscience National Laboratory. We are compiling and developing many methods here to speed up drug discovery in Brazil, especially from Brazilian natural products. And this paper reflects some of the work that we have done, and which is, of course, open for the whole community in the world.
Thank you very much. That's fantastic. Right. The first question I would like to ask you, Raphael, your graphical abstract, it presents a really nice summary of the work. Could you please talk us through the graphical abstract and by extension, the paper in simple terms?
Yeah, sure. In this work, we use the high-throughput crystallography, especially protein crystallography, as a tool to identify ligands, in our case, from complex mixtures of natural compounds. So the idea is to use natural crude extracts or fractions as source of molecules to directly perform the soaking or crystallization with a target of therapeutic interest. So we use the protein crystal as a kind of hook to capture the bioactive molecule from this complex mixture, and in this complex mixture it can contain hundreds of molecules. So once we identify the electron density, it will give us a structural starting point, or at least the shape of the natural product that was bioactive, that has bioactivity. So what we do differently is the integration of the crystallography with mass spectrometry data to refine our understanding of the bioactive molecule that we identified. So combining crystallography and mass spec data, we're able to make more accurate proposals or even completely identify the structure of the bioactive compound. So the advantage of our pipeline is that we can capture the bioactive molecules in very complex mixtures, such as crude extracts or initial fractionation, and do it fast and robustly using synchrotron beamlines like MANACÁ.
Brilliant. Thank you very much.
Well, it's my turn. I would like to start off by saying that I'm so happy to, have you guys here, as a Brazilian, and having the authors of this beautiful paper, as Brazilians as well. So my question goes to Daniela. Dani, what do you think your approach is applicable around the world, or is this something very particular to Brazil because of its huge biodiversity?
This approach is applicable for I think any country. Of course, if it's a country that has a huge biodiversity, it can use for sure. But there are initiatives in the world, even in the US and Europe and Australia, to organize libraries of natural products from the whole world. And the NCI is an example. The Compounds Australia is another example. In France, we also have a chemical library. So we can once you have a chemical library, you can use this approach, especially if the compounds are not purified. So this is the main point. I think this approach is helpful because you can directly screen in bioassays these samples, these chemical samples, and you can then use the heat samples directly with crystallography in a mixture and then capture the compounds. We did that for natural products, of course, because you are working with natural products. In Brazil, you have huge biodiversity, but you can also use these. The natural product community is now one of the main things that we are developing in many countries in the world, is to use microorganisms to produce compounds, even heterogeneously. And then we have also a mixture. And then you can use this approach also to capture the bioactive compound. So it's another way to go. And in terms of microorganisms, all countries have many microorganisms that are not already identified and that produce many compounds that can be used that can use this approach to accelerate the discoveries. And also, this approach can also be used in chemical, synthetical chemistry pipelines, in which we are synthesizing molecules in different scales, and also in mixtures. Then you can also use this pipeline to capture the bioactive compounds, and also cocktails of fragments and so on. So everybody can.
Yeah, very interesting. The idea of using mixtures of compounds and filtering, the ligands out. Very interesting. So my following question is to Andrey. Andrey, what makes the Sirius synchrotron and the MANACA beamline special in terms of equipment?
Thanks, Chris. For me, it's a pleasure also to be here and for the invitation. Well, Sirius is a 4th generation synchrotron, and it's the brand new Brazilian synchrotron. And as a 4th generation synchrotron, it was built to provide a very small and very intense X-ray beam. And crystallography, mainly protein crystallography, can take advantage of that to diffract like challenging or small crystals, which is very good for structural biology. This already opens a lot of opportunities, mainly in our region that don't have much large-scale facilities like that. But from the beamline point of view, we are also trying to automate this process a lot, because it's important for these large scales, screening scale experiments. And I think this combination of a very good X-ray beam with the automation at the beamline can create a new environment for protein characterization and drug discovery. And I believe that also this automation can help another users that are not very used to these tools or used to this data processing in crystallography to access this technique. And of course, regarding the paper, I think this type of collaborations with users like Daniela and Raphael and his team help us a lot to develop these new tools and specific tools for specific experiments. And we hope that these very highly automated methods can become the standard for the beamline very soon and be available for all the users.
As a user, and I strongly support all our researchers in the field to collect data at MANACA. John?
Thank you. My next question is for Raphael. So I'm a software person, And I think our viewers were interested to learn a little bit more about the software pipeline that you are using. How much of it has been developed in-house, and what tools are you using to connect all the different pieces?
Yeah, our pipeline is basically divided into 3 stages, starting with the automatic processing at the MANACA beamline by the MANACA autoproc, and followed by data preparation, which includes the 4 scripts that we described on the paper. This script is used to the resolution cut, the molecular replacement, and to calculate missing reflections all automatically. And finally, the data analysis is performed with Panda, a very nice algorithm developed by Nicholas Pierce to discover low occupancy ligands. So the 2 initial stages were developed entirely in-house. And once the data is prepped, the Panda analysis runs in a cluster here at the Sirius, in a cluster machine in Sirius. And I think it's worth mentioning that we are integrating the scripts with the MANACA autoproc with help the Andrey's team to do it more fast and more automatically. The MANACA users can collect their data and will be automatically processed and ready to be analyzed by Panda. And Andrey, if you can comment with the MANACA auto process point of view, what they do, what the scripts do.
Yeah, as Raphael said, there's a part of the processing that was made offline that time, in the time of the paper, that we have now also incorporated at our pipeline at the beamline. But just a step back, here at MANACA, we have developed mostly the entire pipeline in-house. But of course, using a very well-established crystallographic software like XDS and CCP4 package, also Phoenix package. And we try to integrate all this process using mainly Python scripting. But not only that, I mean, the most of the background language is Python scripting, but we also trying to develop some web servers and websites to users access the data, start processing, visualize the data. Again, with the idea to have this very easy access to the data for non-specialists. So the idea, I think we are in the way to do that, but I hope very soon we have like just one button to be pressed, and then we get the data processing and the entire pipeline running. Again, I have to stress out the importance of this type of collaboration, because we was developing these things at the beamline, but these problems, these questions from the users put some pressure on us to make this fast and make it this in the time that is important, and also the help from Raphael with the data processing help us a lot to integrate all these things and make a data processing pipeline that fits our needs and make possible to process this type of the data.
Thank you very much.
Okay, so my next question goes to Dani, but everybody can contribute. How might this methodology, now we're speaking to the screening of ligands, influence collaborations between structural biologists around the world?
Great question. And I think it also goes with one of the main points of the techniques, which is to use crystallography as a tool nowadays. Né? You can now, through high throughput crystallography, like shooting 200 crystals a day and processing without human interference, at least from the beginning, you can do that. You can use crystallography as a tool. So I think this type of methodology is not only helping in structural biology collaboration between among structural biology teams, but also of structural biologists with other scientists of other fields, as natural products, medicinal chemists, and even others that you can imagine. We can with all this pipeline set, we can attract more users of other fields for crystallography, and we can expand, widen the connections of the structural biologists with other groups and other scientists and other types of research that we can do. One thing that I just want to mention is regarding the method itself that we described, is that we don't need to purify the compounds, and this is a bottleneck in many fields. Natural products, for sure, that we have to purify the compound. For synthetic chemists, also one step more that you have to do. And using crystallography to do this bioactive compound capture, or ligand, at least protein ligand capture, it's very important, and I think is something that opens many venues for using crystallography, especially in drug discovery, but not only. We are now in a moment in which in drug discovery we're very looking forward allosteric ligands of proteins. And sometimes it's difficult even to develop a high-throughput screening assay for that, or something that's more indirect. And using crystallography for capturing these allosteric ligands is for sure one thing that I think this type of methods can really contribute, and to bring new ideas to develop molecules for specifically binding to proteins and specifically modulating the protein activities. We have just to tell about some programs that we are running now here at CNPEM, which are open for the whole community. I want to highlight the bank of molecules with this one program that we started last year, and we are inviting our researchers from all the world, the whole world, and especially from Brazil and Latin America, to deposit natural samples or synthetic molecules here that we can organize, manage the compounds, and track all the activities that are done, and then expand the collaborations and the things that can be done with these compounds, including screening at different bioassays, using different protein crystals to investigate if these molecules can bind to. And this is an open program that everybody is invited to send the samples. And also here at CNPEM, we have many open facilities. We just opened the high-throughput screening facility, so everybody that wants to do high-throughput screening assays here can also bring the molecules or the targets to screen with the bank of molecules and vice versa. And also, probably Andrey would like to comment how we operate in a very collaborative approach in CNPEM, just having even having the open facilities as MANACÁ, but also doing the collaborations and trying to connect researchers from different fields to push research and innovation here in Brazil.
Thanks, Daniela. That was great. A good overview of what you guys are doing. Thanks, John.
Thank you, Chrissy. My last question goes to Raphael. Were there any challenges associated with the processing and deposition of large amounts of data?
Yeah, I think that the most bottleneck is the data deposition, because if one would like to reproduce the results, all the data will have to be deposited in some data bank, like PTB, for example, including all the ground state data sets used to calculate the average electronic density, all the potential bound data sets. So depending on the size of the crystallography campaign, it can reach like thousands of data sets to be deposited. So I think that's the most bottleneck.
So we would like to thank Daniela, Raphael, Andrey for the interview, for the very interesting paper. I'm sure the community will be very interested in listening to your work in more details. So thank you very much, and see you in Brazil.
Thank you very much.
Thank you.
Muito obrigada.
Thank you, Cryst. Obrigado.
Thank you, guys.